Are ya ready, kids?
Aye, aye, scientist!
I can hear you!
Aye, aye, scientist!
Ooh! What must the cell do before dividing?
DNA, replication!
And what do you think this process is called?
Semi-conservative, semi-conservative!
What's needed for it to occur properly?
Enzymes, enzymes!
In what direction happens it possibly?
5' to 3'.
Semi-conservative enzyme activity. 5' to 3' DNA replication.
Second verse.
What is the enzyme that unwinds DNA?
Helicase, helicase!
And how does it separate antiparallel strands?
By breaking the H bonds,
So why does it do this and not just rest?
so both strands can be used.
And what is created by this separation?
Replication fork, replication fork.
Helicase breaks bonds, creates replication forks. Helicase breaks bonds, DNA replication.
Third verse.
Before produces a lot of tension.
That's true, that's true.
What enzyme attaches to relieve this?
Topoisomerase, topoisomerase.
And what protein keeps the strands separated?
SSB, proteins.
And how is the separation crucial?
So the DNA doesn't rewind.
Topoisomerase, SSB proteins, tension and separation, DNA replication.
Fourth verse.
What happens when these strands are separated?
DNA polymerase III goes to work,
And what is it that the enzyme can do?
adding DNA nucleotides.
In what direction are the nucleotides added?
5' to 3'.
And what is resulting from this addition?
Elongated strands.
DNA polymerase III adds the nucleotides, elongates both the strands, DNA replication.
Fifth verse.
Where does the polymerase add the nucleotide?
To a free 3' OH group.
But where's the VOH originate from?
RNA primers.
What are all of this RNA primers?
Store RNA nucleotide sequences.
How do the primers get on DNA?
RNA primase.
OH and primers added by primase, RNA nucleotide, DNA replication.
Wait, can we pause for a second, scientist?
Sure thing, student.
Is it true that DNA can only be synthesized in a 5' to 3' direction?
Yes, that is correct.
But since DNA is anti-parallel, the template strands would be going in different directions from the origin of replication. So how can it both be synthesized towards the replication fork while maintaining the 5' to 3' directionality?
Well, the process involves something called Okazaki fragments. On the lagging strand, DNA is synthesized in parts because it can't be synthesized continuously if it wants to simultaneously be synthesized with the leading strand.
Oh, I get it now. But doesn't that mean there's a leading and lagging part of both strands going in each direction from the origin of replication?
That's right. Let's continue the song and talk about fragments some more. How many primers are on the lagging strand?
Multiple, multiple.
But what can be done about this RNA?
Substitute with DNA.
What enzyme is doing this substitution?
Polymerase 1, polymerase 1.
And what's the product? Tell me now and move on.
Only DNA, only DNA.
Multiple primers, polymerase 1 fires, RNA replace, DNA replication.
Seventh verse.
But now there is absolutely a problem.
There's a nick, there's a nick.
But where exactly is the nick?
Between Okazaki fragments.
And what enzyme is it that seals these fragments?
DNA ligase.
And what do we have now? It soon ends.
Continuous replicated DNA.
Move the nick sealing, ligase leaving, continuous strands, DNA replication.